Welcome to Gibco Education's stem cell reprogramming journey. Today, we're going to look at induced pluripotent stem cells, also known as iPSCs. iPSCs have revolutionized the field of stem cell research by simplifying the generation of donor-specific stem cells that can then be used for disease modeling and basic and cell therapy research. Skin and blood cells are commonly used to derive iPSCs because samples are relatively easily accessible and they can be expanded into large populations. Conversion of somatic cells to iPSCs requires expression of transcription factors such as OCT4, SOX2, KLF4 or c-MYC, because these genes are not actively expressed in somatic cells such as the fibroblasts seen here. To date, different sets of reprogramming factors and gene delivery methods have been tested with varying levels of efficiency and safety. The first method utilizes retroviruses or lentiviruses, which require integration of the viral genome in the host chromosome to express reprogramming genes. Normal gene transcription allows the inserted transgenes to be expressed along with the host genes, which is necessary for the transition to an iPSC. However, integration of viral DNA into the host genome disrupts the genome of the cells and can cause insertional mutagenesis, which can render the iPSCs and downstream derivatives unsafe for use. Another method of reprogramming is via episomal vectors. Once transfected into cells, these DNA vectors reside within the nucleus, but are designed to be non-integrating. A third option is the Sendai virus. The Sendai virus is a single-stranded, negative-sense RNA virus that replicates in cytoplasm. This means it does not enter the nucleus and consequently does not integrate into the host genome. The Sendai virus replicates independent of cell cycle, producing very high copy numbers of the transgenes. Once iPSCs are established, the virus will dilute out of the cells, leaving behind no footprint. In the case of the Invitrogen CytoTune iPS Sendai reprogramming kits, 3 Sendai vectors, KOS, KLF4 and MYC, express the 4 Yamanaka factors, OCT4, SOX2, KLF4 and MYC. These viral vectors are added to the dish of cells and incubated overnight to begin the reprogramming process. Expression of exogenous transcription factors eventually leads to endogenous expression that is characteristic of pluripotent stem cells. As iPSC colonies form, it's important to consider the maintenance medium used to support them. Gibco Essential 8 media contain no BSA and therefore support high reprogramming efficiency. Gibco's StemFlex medium has been shown to increase colony size compared to traditional media, making identification and picking easier and also offers an every other day feed schedule, reducing hands-on time. iPSCs have revolutionized the field of stem cell research by simplifying the generation of donor-specific stem cells that can be differentiated into 3 lineages for downstream applications, including mesoderm, endoderm, and ectoderm, such as the neurons you can see now. While you continue to push the boundaries of what is possible in the world of stem cells, the quality of your cells, PSCs and beyond, depends on robust media and reagents that work together for today's stem cell challenges. Make Thermo Fisher Scientific your partner of choice today.